Hey all, welcome to ShareTreck. This is Raj here. In today's video, I want to talk to you about some risks that I had mentioned in my earlier videos. If you recollect, I was quoting a few reports here and there that were talking about potential risks with double-strand breaks due to CRISPR-Cas9 usage. Today's video is going to be a positive video in the sense that it's going to talk about some new developments that give us hope that our genomic investment is going to be less riskier. So let's get started. Welcome back. So the news article is as follows. The scientists from Institute for Research in Biomedicine led by Dr. Frank Supek have now reported that depending on the targeted spot of the human genome, CRISPR gene editing can give rise to cell toxicity and genomic instability. This unwanted effect is mediated by the protein p53, which is a tumor suppressant protein, and it's determined by the DNA sequence near the editing point and various epigenetic factors in the surrounding region. p53 is a protein known as the guardian of the genome. We have spoken about this earlier. It detects DNA damage and leads the cell to stop dividing and can cause programmed death, thus preventing them from reproducing and expanding the mistakes in their DNA. Therefore, p53 underlies a natural protection mechanism against cancer and other DNA-related complications. CRISPR gene editing often requires cutting both strands of the DNA. In some cases, this manipulation can trigger a p53 response in which edited cells can be tagged as damaged and are then eliminated by p53 action, thus reducing the efficacy of the gene editing process. This particular report explains that the main complication regarding p53 and gene editing is that cells that overcome CRISPR editing might do so precisely because of defective p53 functioning. This means that these CRISPR-edited cells may be less able to detect future DNA damage and or tag cells for programmed death. As a result, the gene editing procedure could end up favoring cell populations that have unstable genomes, meaning they are prone to accumulating further mutations, thus increasing the risk of developing malignancies. So whatever was reported earlier as toxicity of double-strand breaks due to CRISPR-Cas9 is now much more focused, and we have a specific instance where things could go wrong and the reasons why. Using advanced computing tools, the researchers in their genome data science lab have analyzed the most popular CRISPR library designed for human cells and have detected 3,300 target spots that show strong toxic effects. They also found that around 15% of the human genes contain at least one toxic editing point. While we wait for this report to be peer-reviewed and confirmed, we as genomic investors can try to understand what this means to genomics industry. This, in my opinion, is a positive development. Now there is a reference library of edit points that are toxic, and therefore any genomics company can validate against this library before investing in any therapy in their pipeline, knowing in advance that the edits could be problematic. This could potentially mean that some conditions may not be resolved through gene therapy if they fall within those 3,300 edit points. But at the same time, this reduces the risk around progressing a gene therapy, investing lots of money and time and effort into it, and going through the FDA process only to discover towards the end that there are issues that can potentially derail the entire therapy. Now these companies can validate the edits against the table of toxic edits and thereby reduce the possibility of failure due to toxicity. It is still too early to celebrate, but overall this industry is moving in the right direction, and I'll keep track of this for us and bring to you any more information that I get going forward. I thought that I would like to highlight this to you, that there is a lot of research going on to further refine CRISPR-Cas9. As you saw, the whole history of this industry, right from the introduction of CRISPR-Cas9, we have been refining the process. We have come across base edits which avoid double-strand breaks. And now this new development with the identification of 3,300 points where edits could have toxic effects. All this can only improve the outcome of any gene therapy that is in progress, and also help companies, genomic companies, to avoid investing in therapies that could potentially fail in the FDA clinical trials due to toxicity. With that said, I hope you liked this video. Please press a like out there if you did. And if you're not yet a subscriber, Please subscribe and help us grow this channel. Subscription is absolutely free. Thanks and have a great day. Bye for now.